have the pleasure of introducing Ken Anderson from the Dana-Farber, who's going to tell us what's new in myeloma.
Great, thank you very much, Bruce, and it is an honor to be here. Here are the objectives of the talk. Very quickly, I'm going to just mention the perspective here on myeloma that many of you know, but it really wasn't treatable till the 1960s when melphalan and prednisone came, and then as we moved into the more recent times, the 80s, high-dose therapy with marrow, and then in the 90s, high-dose therapy and stem cell transplant. But I start with this just because since really 1998 onward, the whole paradigm for treating this disease has changed. And we have the concept of targeting the tumor in the microenvironment, which is what I'll be talking about here today. And we have novel agents, or new agents at the time, proteasome inhibitors, immunomodulatory drugs, that could, in vitro and in vivo assays, overcome drug resistance, so-called cell adhesion-mediated drug resistance that frustrated conventional therapy. And as all you know, and many of you in the audience participated in these trials, the drugs went in advanced disease, relapsed disease, and upfront, and in record time were approved. And the neat thing that happened, which is really what this meeting is all about and why we're all here, is did we help patients? And the fact is that we've doubled the survival together. It's now 3 to 4, up to 7 to 8 years. And there is much reason for optimism in the future. So the concept is when we do the genomic analysis, as we and you all are doing, of the tumor, that's important, but it's only part of the story. Because by virtue of cell-cell adhesion and also the induction, in this example, of cytokines in the microenvironment, the genetic program and functional sequelae of the tumor cell is actually further complicated, and you really need to overcome all of those growth, survival, and drug resistance advantages when treating a patient. So here's an example. There's the evil myeloma cell there binding to the bone marrow stroma, and this is a transcript study showing the different transcripts that are upregulated, one of which in the red is the ubiquitin proteasome system. I guess we can point up here. In fact, that's at a transcript level. If you look actually at a protein and a functional level, the chymotryptic, tryptic, and caspase-like activity of the proteasome is also upregulated. So in more plain English, the proteasome inhibitor is actually a better drug, it's a better target when the tumor is in the microenvironment than when you study the myeloma cell ex vivo. So this is a very important concept. Now this cartoon I recently made that summarizes the activities that bortezomib, the proteasome inhibitor, induces in myeloma cells. And there's some direct effects here on growth and survival. There's effects, after all, targeting the chymotryptic activity of the proteasome. There's effects on cell cycle proteins. There's direct induction of apoptosis that's junk mediated, and ER stress induction. But the reason I put this up here is that it's targeting the tumor and the microenvironment. Very important to remember when we talk about targeted therapies, because as shown here in the middle, only appreciated after the drug was approved are its effects on enhancing osteoblast function and inducing apoptosis of osteoclasts, and thereby helping the bone complications in our disease. It's also anti-angiogenic. But the point is, it is overcoming the advantages that the microenvironment confer to the tumor itself and acting on the same circuitry to have different sequelae in the microenvironment. Now, going forward, we're trying to figure out how we can use bortezomib better, so I want to just spend a minute on that. And one example, as has been talked about here, is biomarkers or predictive tests that might tell us who among patients with cancer might respond to a proteasome inhibitor. Right now, myeloma and some lymphomas are the main patients benefiting. So here's an assay that it's not commercial yet, but in fact there are thoughts that we can try to make it more user-friendly in that regard. But it looked at cancer cell lines and cancer patient samples, and it simply looked at those tumors that had, relatively speaking, a low proteasome capacity for the load that was placed on the proteasome. That's the classic myeloma cell that has large amounts of ubiquitinated misfolded protein. So it's easy on the right-hand side of this slide to overcome that with a proteasome inhibitor and have an effect. Contrary-wise, in many solid tumors, the opposite is true. The load on the proteasome is really not very much relative to the capacity, and this proteasome stress index, as we called it, may someday be useful as a biomarker. But as many of you know in the room, even when we first put this drug in the clinic together back now almost 10 years ago, I think it was kind of neat because it was one of the first studies in which profiling was done on all the patients, or we tried at least on all the patients, to try to see if we could get a signature of who responded or not. So in the upper left-hand corner, in the red, the transcripts that were overexpressed in responders versus the lower right, transcripts overexpressed in those who didn't. And I mention it to you because this may someday, and similar analyses, allow us to have a signature of response, but already it's helped us design and inform clinical trials to use this drug better. So here's one example I'll just show you. HSP27, heat shock protein 27, at a transcript and protein level was induced in patients whose myeloma became resistant to bortezomib. So we brought that observation from the bedside back to the bench into the laboratory, and we knocked down, in this example, heat shock protein 27 in a Bortezomib-resistant cell line, and in so doing conferred sensitivity. We did the opposite. We overexpressed HSP27 and conferred resistance. So that and some other work was the basis, as is shown here, for a clinical trial. So to my knowledge, there still isn't a heat shock protein 27 inhibitor, but Teru Hiroshima and our group showed very nicely that if you downregulated p38 MAP kinase upstream, you could downregulate heat shock protein 27, and in cell lines and in patient samples, in so doing, overcome bortezomib resistance. So this informed the design of a clinical trial of a p38 MAP kinase inhibitor with bortezomib. I think the best example of preclinical modeling translating to the bedside, and I believe making a huge difference, is this one. Bortezomib, as we all know, the proteasome inhibitor, does trigger apoptosis. It's mostly caspase 9 greater than 8. And one of the other novel drugs, lenalidomide, is mostly caspase 8. There's a lot of reasons to put these two drugs together, but dual apoptotic signaling is one. And if you use these combination in the clinic in patients whose myeloma is resistant to either agent alone, 58% of the time you have a response. And if you use them up front, bortezomib, lenalidomide, and dexamethasone in newly diagnosed myeloma, only 100% of people respond. 3/4 get a very good partial response or better, and half of the patients get a complete or a near complete response. And honestly, those are unprecedented numbers, as many of you in the room know. They're so unprecedented that we're just started what I call an intergalactic clinical trial. It's with our French colleagues, and the concept is very simple. 1,000 patients with myeloma will get this exciting combination novel agent therapy. All will get stem cells collected, and then half will get a transplant and half won't. And we'll learn whether a transplant adds to the high response extent and frequency here, or we'll learn about the durability here of novel therapies. This trial has a P01 around it now, and actually very nice science, including whole genome sequencing at the time of diagnosis and relapse. So we'll learn a lot about myeloma with that trial. So now I just want to spend the last part of my talk telling you about where I think we're trying to move in terms of the future. And it's quickly going to be in 4 ways: personalized medicine in myeloma, novel agents that may be better than the ones I've mentioned already, a little bit about our thoughts on immune, and then again using science to inform combination therapies. So quickly, as has been apparent at this meeting, there is obviously a move to personalized medicine, and it's at many levels. I'm just put in a couple of slides here to show you unpublished data from our center, where we looked in this example with just SNP array DNA analysis to look at responders versus non-responders, which we're now doing for most of our protocols. And this just shows you here extra copy number of 1Q or 5Q and decreased copy number of 12P that correlated in this example with outcome, as is shown here in this population. This happened to be transplant, but this is now being done in novel treatments with all of our protocols. The important point I wanted to mention here very quickly is when we talk about personalized medicine, as we all are doing, We think about it at the time of diagnosis, but we need to think about it at the time in long-term follow-up relapse as well. Because this just shows you two examples from our center of analysis, early tumor versus late. And in the interest of time, I'll just say there are changes here, that there are evidences of abnormalities present early that aren't there late, or there's the acquisition of changes late that apparently are not detectable early. So the concept is, I'm afraid, that we're going to need to do these analyses once we learn how to do them better sequentially over time. Now quickly, what about the next generation of novel therapies? This is the Evo myeloma cell. As many of you know, I refer to this as a New York Yankee very commonly. But in any event, what I'm trying to convince you of is that if you're going to win the World Series, you can potentially target the tumor cell surface. In my model here, you could target in the microenvironment, for example, cytokines, or when you use targeted therapies, think about hitting the target, but hitting the target not only in the tumor, but in the microenvironment as well. So I just put one example of just a cell surface target. So this is an example of a bedside, back to the bench, back to the bedside example. So clinical analysis showed that CS1, an NK cell antigen, was universally expressed on patient cells with myeloma in a clinical analysis. That was brought back into the laboratory. This immunoblot shows you that the proteins there are on sensitive and resistant myeloma cell lines. And here's a monoclonal antibody, humanized antibody against this antigen, CS1. It's now called Elotuzumab. And that and some more science led to a clinical trial. Well, the clinical trial showed only stable disease. And those of you who are from industry in the audience will know that stable disease is not enough to convince people to go forward and develop a drug. So you would say, oh, that's the end of the story, but not so fast, because the laboratory showed that if you added lenalidomide, one of these other drugs I mentioned, to the antibody, you could, in so doing, increase antibody-dependent cellular cytotoxicity in the preclinical studies. So that led to an ongoing clinical trial, combining lenalidomide with a humanized antibody, and so doing to augment ADCC. And as I have in the yellow here, 95% of patients with relapsed myeloma responded to the combination, whereas we only had stable disease when we didn't use the combination. So very exciting data there. Now, what about the proteasome? Let me just share you some new thoughts that I have about the proteasome. Obviously, Bortezomib, as many of you contributed to, opened up a whole new area of research. And so we have now upstream of the proteasome deubiquitinating enzymes that block the deubiquitination, which needs to occur in order for the protein to bind to the 20S core. We have other inhibitors that are selective or not, and we have immunoproteasome inhibitors. So here's an example of a DUB inhibitor, deubiquitinating enzyme inhibitor. And this is just dose-dependent cytotoxicity here on a variety of myeloma cell lines, and this is inhibition of myeloma cell growth in one of our xenograft models. This is actually an oral agent that's coming to the clinic soon. In terms of chymotryptic inhibitors, like Bortezomib, there are several. This one is from Cephalon, known to some of you well. Here's a dose-dependent decrease in cytotoxicity, and here's synergism demonstrated in this right panel here with dexamethasone. Perhaps the furthest along in terms of the chymotryptic inhibitors is known to many of you, called carfilzomib. It's a different structure as shown here. There's no boron in here, as you may notice. And it's deeper and more prolonged inhibition of the proteasome than Bortezomib. This is just one slide of clinical data showing that about 60% of patients with relapsed myeloma who have not yet received Bortezomib do respond to this new proteasome inhibitor. So absolutely, the concept of inhibiting the chymotryptic site for clinical benefit is really validated. Millennium has a second, this is an oral inhibitor, also from Onyx, the same thing, oral proteasome inhibitors that are chymotryptic targeting are coming. This is, in fact, myeloma cell death here of patient cells, large amount, a very potent agent. Millennium has an oral chymotryptic inhibitor that has different biodistribution than Bortezomib, and in the animal models can overcome Bortezomib resistance. We and others in Partners System have this in clinical trials in the Dana-Farber Harvard Cancer Center. But here showing you that the degree of proteasome inhibition is very similar to bortezomib, and also we have activity against patient cell lines and patient cells. And there's a more broad inhibitor. Chymotryptic inhibition works. What about chymotryptic, tryptic, and caspase-like inhibitor? Here's an NPI-0052 inhibitor that has more broad inhibition that, in fact, in our preclinical models, inhibits myeloma, human myeloma cell growth in a xenograft model and cures the mice. And Paul Richardson at our center has reported that this more broad inhibitor in very far advanced relapsed refractory to bortezomib myeloma can induce, even in a phase 1 trial, 58% stable disease. And finally, what about the immunoproteasome inhibitor that I had on the slide? Well, the immunoproteasome may very well be a good target in myeloma and other blood cancers because normal cells have much less in terms of the load of immunoproteasome compared to malignant cells. So here is very exciting data that shows dose-dependent inhibition of growth here, and in fact, no effect on peripheral blood mononuclear cells whatsoever. So this immunoproteasome concept may be coming. And this is in vivo again, inhibiting myeloma cell growth, prolonging survival of the mice, inducing apoptosis in the tumor, anti-angiogenesis here, decreasing proliferation, and another measure of anti-angiogenesis. So inhibiting the ubiquitin proteasome system in different strategies is here to stay. What about the immunomodulatory concept that lenalidomide and thalidomide really pioneered in myeloma? And this, as you know, is a very famous cartoon first drawn by Nupur Raji, who's now at MGH and a myeloma director. But in any event, the direct effect on myeloma, but the reason these agents like Velcade, the proteasome inhibitor, are different, they're acting here on the microenvironment, and they're immunomodulatory because they upregulate the immune NK cell, T cell, and NKT cell activity in patients on the drugs against their own myeloma. I share it this morning quickly because I think known to many of you, pomalidomide is more potent, and in fact, in the laboratory studies, overcomes lenalidomide resistance. Apparently in the clinic it does as well. We have very high response rates here, even including myeloma that is resistant to bortezomib and lenalidomide. And this slide I just put in because if I can press it enough times here, we have been talking about targeting the tumor, myeloma, and what are the effects on the microenvironment. Well, this slide shows you a summary up to and including Velcade, if I press it enough, there you go, where, in fact, if you target the bone disease in myeloma, you can, in so doing, this time affect the myeloma cell. And the example I just want to show you is this one. It's called DKK1. Myeloma cells secrete DKK1, which suppresses osteoblast function. Based on that and a lot more science, antibodies to DKK1 have gone into the clinic in our center. And these antibodies, if you add DKK1 antibody, you can restore new bone formation in this in vitro assay. But importantly, in our preclinical studies, DKK1 antibody, which is called BHQ, can not only restore bone formation here, but it inhibits myeloma cell growth in 2 different models of xenograft human multiple myeloma. So we have an ongoing trial where we're looking this time at a primary endpoint in bone, but in fact seeing effects also against the myeloma. A couple of just closing thoughts here. So we talked about the analysis for personalized medicine. We talked a little bit about novel agents. We're also excited about immune therapies in our disease, and that promise has not been delivered, as many of you know. Here is just a cartoon showing you many abnormalities that we and others have found in multiple myeloma. Some of them are translating. So, for example, there are antibodies to Th17-associated cytokines that are coming into the clinic to try to correct abnormalities. But the reason I show it to you today is that genomic analysis, I think, is going to make a huge difference in our approach to immune treatments. And the reason is you can do genomic analysis and define genes and gene products that would be on the cell surface and would be decent targets for an immune treatment. But then you can also look and ask, of these gene products, which of the peptides that are composing these gene products, and in this analysis we found 9 gene products here, which peptides would be presented in the context of patients of particular HLA types. So more plainly, the genomics is being used to define the target and the peptides, and it's also being used, genetics, to define the appropriate host. So I think we have opportunities here now that we haven't had before to deliver on the promise of immune therapies. And now just to finish with the last topic, which is combination therapies. Don't have to tell anybody in this room that that's really, combination therapy is the only way we've really made a major difference with traditional chemotherapy. But let me just show you some examples of why I'm excited for the future of how the preclinical studies can actually inform clinical trial design. So here's studies from our lab showing that bortezomib can induce caspase-mediated apoptosis of myeloma, but in fact, there's an induction of AKT activity. And if you abrogate that activity with perifosine, you get synergistic killing in the laboratory. So that and a lot of other science went into the clinic, bench to the bedside, and here we have a compilation of phase 1-2 trials showing that adding the AKT inhibitor to block the induction of AKT by bortezomib gives you responses from 30 to 50% here that are durable and actually have led now to a phase 3 clinical trial of bortezomib versus bortezomib and the AKT inhibitor for registration. As many of you know, my most exciting example quickly is this one. Remember, bortezomib blocks the breakdown of ubiquitinated protein here, and in fact is active in myeloma. We and others have been obsessed with studying this other pathway called the agrosome autophagy pathway for protein degradation. And if you block both pathways, you get the most synergistic killing we've ever seen. If you just focus right here for a minute, the darkness of these bands is the amount of ubiquitinated protein in a myeloma cell. If you block HDACs, here, if you block the HDAC inhibitor rather to block the agrosome here, bortezomib to block the proteasome here, and if you add them together, you get synergistic killing, as is shown down here, with a massive accumulation of ubiquitinated protein. So this has translated to the clinic, and with our Spanish colleagues, we actually have studied this, and panabinostat, a very broad HDAC inhibitor, in this example used to block the agrosome, with bortezomib to block the proteasome, 70% response in advanced myeloma, including 60% response in phase 1-2 trials in bortezomib refractory disease. Now, one of the neat things that's happened at our cancer center is we have the ability to do synthetic chemistry now that we never had before. So if you wanted to block the aggregosomal system for protein degradation more selectively, you would block histone deacetylase type 6, because histone deacetylase type 6 binds to ubiquitinated proteins and binds to the dynein motility complex and moves it to the agrosome for its degradation. So in the laboratory, Jay Bradner and colleagues actually have made us a HDAC6 selective inhibitor. The assay is, does it in fact cause accumulation of acetylated tubulin? And it does here. As you can see, the tubicin was the prototype. Here is the drug, almost medicine here, inducing selective increase in acetylation of tubulin. It's not here, but you do not cause increased acetylation of histones. This is unpublished data. So this is bortezomib, the proteasome inhibitor. This is carfilzomib. Remember, I said if you block both pathways, it would be the synergistic killing. Well, here's tiny doses of bortezomib, and you can see dose-dependent killing of myeloma cell lines here by the lab and by the clinical reagent for both of these proteasome inhibitors. That's cell lines. Here we have patient samples inducing the induction of acetylated tubulin in patient samples with a huge, very tiny doses of bortezomib here, huge evidence for synergistic death. And I'm very proud to tell you that within our system, this drug will have gone from the bench about a year ago into the clinical trials arena early next year, in almost right around 1 year. In any event, the final point I want to make is now we have all these options. How are we going to combine them? We can't possibly do all the different combinations. Can't we make sense of this? And I just wanted to show this last concept. This is the work of Konstantin Mitsiadis in our group, but he's developed a high-throughput assay where you can plate here the microenvironment, stromal cells, for example, and you overlay them with bioluminescent tumor cells, and so you can look at 1, 2, 3, or 4 drugs across dose ranges and sequences. And here's the important point. Remember, here's survival of myeloma cells. Decadron can induce cytotoxicity in the absence of stroma, but with stroma, it's completely attenuated. Down here, in fact, bortezomib kills cells equally well in the presence or absence of stroma. So we're using this assay quite routinely now to define new targets, to validate new targeted agents. And the last slide I just wanted to show you is recent data. But this is a drug screen. Here's a bunch of targeted therapies here along the x-axis, survival of myeloma on the y-axis. If, in fact, the survival of the tumor is equivalent in the presence or absence of stroma, it falls on this white line. If it's on the orange line here, this means more survival of myeloma when it's in the stroma. We're not choosing such targets or targeted therapies. But excitingly, there's going to be a population of cells illustrated here where the target is only exposed in the context of the microenvironment. I think that represents a whole new area of targets that we haven't been able to appreciate previously. So what I've tried to do, albeit very quickly, is just telling you that it's been a very exciting time in myeloma. We're now able to achieve responses at an extent and frequency, and it's translating into progression-free and overall survival advantage that we haven't appreciated. And I tried to show you that we can use science to not only define targets and targeted therapies, but honestly to inform the design of clinical trials that are much more likely to be positive. And then I showed you at the end the way we're trying to define perhaps 3 and 4 or more drug combinations as needed using this science. And as many of you know, I affectionately call my team the United Nations Against Myeloma. This is the laboratory that currently is here. These are the flags. It goes from bench to bedside and back in order to define new targets and targeted therapies and get the next generation, inform the next generation of clinical trials. So I think I'll stop here, and thanks very much.
Ken, would you take some questions?
Sure, sure. Any questions? Oh, there's one back there, I think.
Thank you very much. In regards to the earlier presenter before the coffee break, in regards to these combination therapies, prior to approval and kind of, trying to prosecute clinical designs that way. I'm wondering, if you can weigh in with your vast experience in terms of how feasible you think that really is.
No, I think it's, you're talking, I think, about Eric Rubin's very nice presentation, and I know it well. I do think that we are going to need to do it together, that, those of us in academia and in industry to actually help inform adaptive trial design so that we can do this. The plain truth is, and I'll just use myeloma as an example, the only novel drug that got approved as a single agent was Bortezomib. Thalidomide was with steroids, Lenalidomide was with steroids, Bortezomib, Doxil. It's unlikely, in my opinion, that novel agents, at least in our setting, and I think in most, are going to have sufficient activity because the diseases are getting tougher and tougher when they're relapsed and refractory. So we have to be creative on how to sort out benefit and side effects, as one of the questions asked, for novel combination therapies. And I think the FDA has asked for guidance for those kinds of trials. And I do think that that is the future if we're going to expedite drug development. We have to be better about combination therapies. The other thing I'll just mention, and industry is in this room represented very well, but that means cooperation between companies because not any one company necessarily has the right members of the Red Sox. You all have Yankees, but you're not going to, it doesn't matter. But no, I'm just teasing. But it's going to have a new level of collaboration and cooperation. Okay. Yes.
Ken, lovely talk. Can I just ask you, in relationship to the importance of the specificity of the HDAC6 component, what do you think the mechanism is in terms of degradation? Do you think that it's an acetylation-controlled event that's HDAC6 regulated? Is that what you think is going on?
I think that's true. The question is, what are the relevant substrates? And this is a very, very exciting area to me. My lab is pretty obsessed with this now because although these agents are named histone deacetylases, honestly, the substrates are really different. And so we're doing what's called the acetylome, right? Proteome, genome, whatever. So we're doing that, and we're trying to understand, to answer your question directly, honestly, what are the relevant substrates that correlate with clinical activity? It may or may not be what I've shown you. It's just too early to tell, but I think that is the future.
Ken, I'd like to ask a question. Many of the previous presentations talked about addressing molecular lesions in the tumor,
Right.
particularly the driver mutations. In myeloma, there are a variety of driver mutations, or at least separate categories of disease in which specific mutations are found. Is that informing any of the therapeutic attempts at this point?
Yeah, we're honestly just very much in our infancy there, Bruce. I think the best example in myeloma, and you're absolutely correct, it's very heterogeneous, from traditional karyotyping and also genomic analysis is the 414 translocation patients who, by virtue of that, it's about 10 or 15% of myeloma, and they have FGFR3 and also MMSED, but FGFR3 on their cell surface. So as some of you know in this room, that has spawned, Bruce, a clinical trial of an oral FGFR3 tyrosine kinase inhibitor in those patients. We've just completed, together with our colleagues at the Multiple Myeloma Research Consortium in the Broad and TGen, a sequencing study in myeloma. So we found some interesting things we already knew, RAS mutations, but we found some things that we didn't know, like VRAF mutations. So there are going to be, as we always call it, right, low-hanging fruit. There will be some lesions that need to be validated to be driving. None of this is validated yet. But assuming that it does, there may actually already be available some oral agents from other contexts that could help, or agents that could help us.
This would open up a totally new area for therapeutics.
Yeah, I agree. I agree. It's very exciting.
One other thing prior to our next presentation, which is about how one goes about stimulating research from a foundation level in rare diseases. You've been very interactive with the Myeloma Foundation. And I think it would be worth just a word of how important have they been in terms of stimulating and supporting research?
I think it's critical, and John knows this very well. I mean, I think we, you know, as you know, I always make these team analogies, right? It's really a problem. But in any event, but you know, we're only...
You're a team player. That's right.
It's either that or family, but the point is, you know, we're only, there's industry here, there's academia here. We're two very, very important parts of the team that are necessary. You know, one of us can only carry the ball to the 50-yard line. If we're going to score, we both have to work together. But what is the game that we're playing? The game we're playing is to make science count for patients, diagnosis, prognosis, and treatment. So to be honest with you, Bruce, the inspiration for all that we do, but honestly to inform what we do, to participate in the preclinical and the clinical trials, to get sequencing done. Honestly, the relationship that we have to have with patients, and it needs to be an organized relationship in some shape, way, or form, in my opinion, is absolutely seminal to getting progress done.
It's particularly important for neglected or rare diseases,
yes,
where the advocates are not so well organized traditionally, and so that's why it's been a very helpful thing to have Kathy Justy and her folks involved, and others. Any other question for Ken? No, thanks a lot, Ken.